Hello everyone, I'm Mirza Sami, a PhD student from the University of Alabama at Birmingham. I will be presenting our model, BrainGAN, which takes a functional connectivity approach to brain dysfunction prediction. This research was conducted by Yan Lab from the University of Alabama at Birmingham under the supervision of Dr. Yan. This presentation is structured in the following manner. We begin by describing the problem that this research tackles. Next, we will describe the dataset used in this research, after which we'll justify the motivation behind using generative adversarial networks, or GANs. Then, we'll move on to related work, where we will summarize some seminal GAN models, which inspired several aspects of our model. Once sufficient context has been provided on the dataset and prior GAN models, we will provide a high-level overview of our own model. Next, we'll provide a more detailed explanation of the generator architecture, followed by a detailed explanation of the discriminator architecture. After that, we'll describe the procedure used to train the GAN. Finally, we'll conclude by reviewing the experimental results of this model and show that our model achieves a new record in prediction accuracy. Diseases caused by brain dysfunctions are prevalent in the population. For example, autism spectrum disorder, or ASD, is a brain-based disorder characterized by social deficits and repetitive behaviors, and CDC estimates that 1 in 68 children has been identified with ASD. Attention deficit hyperactivity disorder, or ADHD, is a mental health disorder causing impairing levels of inattention and/or hyperactivity, and it affects at least 5 to 10% of school-age children, with annual direct costs exceeding $36 billion per year. Furthermore, CDC reports 5.8 million Americans are living with Alzheimer's disease as of 2020, which is the most common type of dementia. Hence, fast diagnostic prediction of brain dysfunctions is of vital importance. To make brain dysfunction predictions, our model would require brain images as input. Neuroimaging is a discipline within neuroscience and psychology which uses RS-fMRI to generate brain images. There are two major neuroimaging databases, the Autism Brain Imaging Data Exchange, ABIDE, and the Alzheimer's Disease Neuroimaging Initiative, ANDI. We adopt two RS-fMRI brain imaging datasets from these databases to verify the effectiveness and generalizability of our model, namely ABIDE-1 with class labels Asperger's, ASD, and healthy controls. Note, Asperger is a kind of high-functioning autism, and ADHD 200 with class labels Combined, Inattentive, and Control. Note, we can merge the 13 ADHD hyperactive and impulsive patients with the 207 ADHD combined patient into one subcategory, since the sample size of 13 is too small to allow effective training. In such datasets, a brain image is typically parcellated into M distinct regions of interest, and then the pairwise coactivation between them can be represented as a functional connectivity, or FC matrix. It will be described in more details in the upcoming slides. Broadly, the input to our model is a FC matrix, and the output is a prediction of the three class labels for the respective dataset. RS-fMRI functional connectivity is a brain mapping technique that observes changes in blood oxygenation in the brain. It creates what is referred to as a blood oxygen level dependent or bold contrast that can be measured using fMRI. As a standard pre-processing step, 3D and time information from fMRI data are parcellated into M functionally homogeneous regions, and the whole brain FC network is obtained using Pearson's correlation from the corresponding regional mean time series. Note that each element of a FC matrix is a correlation coefficient ranging from -1 to +1. After applying these pre-processing steps, we get a 200 by 200 FC matrix for each subject in the ABIDE-1 dataset. Similarly, for the ADHD 200 dataset, we obtain 190 brain regions, and hence the FC matrix A of each subject has a dimensionality of 190 by 190. In this section, we will justify the need for a generative adversarial network in our model. Both ABIDE-1 and ADHD200 datasets have only around 1,000 subjects, while deep learning models typically require a few thousand samples to train well. Thus, increasing the sample size of such a dataset will have high marginal gain in model prediction performance. Hence, we propose to generate realistic subject samples using a carefully designed domain-specific GAN to improve brain disease prediction accuracy. Having justified our need for using a GAN, we can review some GAN models related to our work. In vanilla GAN, the generator takes some random noise Z and produces fake data that is similar to the ground truth in a database. The discriminator's job, on the other hand, is to discriminate between the real and the fake data. In this adversarial process, the generator learns to produce more and more realistic data, and the discriminator gets better and better at discriminating between real and fake data. The deep convolutional GAN, or DC GAN, is an extension of the GAN model that uses deep convolution layers for the generator and the discriminator. It also uses a ReLU activation in the generator for all layers except for the output, which uses tanh function, a precedence we also follow. The conditional GAN, or CGAN, uses additional information such as class labels to condition both the generator and the discriminator. In our case, we condition our generator with the disease class labels from our dataset. The auxiliary classifier generative adversarial network, or AC-GAN, is an extension of conditional GAN. It adds an auxiliary head to the discriminator to predict class labels. In our case, we follow this technique to predict the disease class labels from our dataset using the discriminator. The information maximizing GAN, or InfoGAN, enforces better structure to the latent space of a generator. It adds more semantic meaning to the latent space by distinguishing between noise and latent code. In our case, we add semantic meaning to the latent space by conditioning our generator with data features such as age, gender, and the label of the disease. This prevents our model from mode collapse, a common phenomenon in GAN training, where the generator produces limited variety of samples. Now that we have described the dataset that we are using and reviewed all related work, we will introduce the architecture of our model. It may be easier to understand this model, one part at a time. So let us begin by focusing on the generator. Inspired by AC-GAN and InfoGAN, we provide data features such as sex, age, and the class label as input to the generator. The sex of the subject is binary encoded, the age is normalized, and the class labels are one-hot encoded before being fed to the generator. Additionally, we also sample some random noise Z from a Gaussian distribution to be used by the generator. The generator takes in all the input data and produces a realistic FC matrix, which is not from our dataset. On the discriminator side, we take either a real FC matrix from the dataset or the fake FC matrix from the generator and feed it into the discriminator as input. The discriminator has two heads. One predicts if the FC matrix is real or fake, and the other predicts the appropriate class label for the FC matrix. With a broad overview of the model architecture presented, let's take a look at the generator architecture in more details. Concretely, we will be referring to the ABIDE-1 dataset in this example, which has 200 by 200 FC matrices. The feature-expanded random input vector Z', which is the input to the generator, is passed through several dense layers to produce an embedding matrix X. This is a 200 by D-dimensional matrix, where each row corresponds to the embedding vector of one of the 200 brain regions in the ABIDE-1 dataset. Mathematically, it can be expressed as follows, where W and B are the learnable parameters. Note, By using d less than 200, the generator only needs to generate a smaller 200 by d embedding matrix rather than the entire 200 by 200 FC matrix A. This allows our model to have less parameters so that it can be better trained with a limited number of samples. Also note, an element AI of the FC matrix A can be generated by taking the inner product of the feature embeddings x sub I and x sub j of two brain regions from the embedding matrix. Thus, the full FC matrix can be generated by taking the inner product of x transpose with x. We use tan-h function in the last layer as suggested in DCGAN to get values between -1 and 1, which is also the range of correlation coefficient of brain regions I and j. We adapt the state-of-the-art FC matrix classifier BrainNet CNN for our discriminator design by extending it to take the age and sex features as input. The BrainNet CNN has special layers. Each has C-simple convolution filters of a particular shape as illustrated here, and performs a specific operation on the brain network to generate an output tensor with C channels. First, a FC matrix A is taken as input and passed through an edge-to-edge or E-to-E layer. This layer does a smoothing operation on the value of an edge based on all of its adjacent edges. Hence, the E-to-E layer takes the inner product between row I concatenated with column j and applies a weight filter to generate a single element ij in the output tensor. We use two E-to-E layers as suggested in BrainNet CNN, followed by an edge-to-node or E-to-N layer to obtain node values by smoothing adjacent edges. Finally, a node-to-graph or N-to-G layer is used to extract a subject feature vector from the node values for final prediction. Once we obtain the subject feature vector extracted from the FC matrix, we transform it along with the subject feature age and sex into the same space for concatenation using dense layers. The concatenated features are then passed into two prediction heads. The validity head does real or fake prediction, and the class head does class label prediction. Each uses a dense layer to obtain a vector of class scores, which are then used as logits to connect to a cross-entropy loss for training. In this section, we'll describe the training procedure for our BrainGAN model. The model is trained iteratively, where each iteration has 3 steps. First, train the discriminator on a mini-batch of real data. Second, generate a mini-batch of fake data from the generator and train the discriminator on it. Third, train generator by freezing the weight parameters of the discriminator. The training curve for our model on the ABIDE1 dataset is illustrated here. The blue curve, named validity real, corresponds to the training accuracy of the real or fake data head in step 1. The orange curve, named validity fake, corresponds to the training accuracy of the real or fake data in step 2. They converge at around 60%, proving that our generator is able to generate realistic FC matrices. The green curve, named class real, corresponds to the classification accuracy on the real validation data for the classification head. It keeps improving, showing that our 3-class classification performance was improving as the model was training. Note, the fluctuations are normal for any GAN training. Here, we will present the results of our ablation study on both the ABIDE1 and ADHD200 dataset. In these tables, GAN referred to our model, and BRAIN refers to the BrainNet CNN model, which only has a discriminator. The hyphenated characters A and S are the age and sex features. These features may be removed during ablation study to check if they're really effective and useful to the performance of our model. The 2-class column heading is the disease or control prediction accuracy, and the 3-class column heading is the 3 distinct disease label prediction accuracy. As can be noted from the tables, using age and sex feature,
enables our model to push the accuracy from around 70% in existing works on a byte 1 to a new record of 73.74%, and we achieved a high accuracy of 72% on ADHD 200, which is higher than the results from the ADHD 200 global competition. We hope these results could provide better diagnostic suggestions at earlier stages of brain diseases.
This concludes our presentation. Thank you for your time. We're happy to answer any questions.